Hello. Today, I'd like to talk to you through our efforts at identifying wheat blast resistance genes, RWT3 and RWT4, that recognize the host species determinants, PWT3 and PWT4 respectively, of Pericularia oryzae. Should emphasize that this is a collaborative effort in which all partners have played essential roles. We have two groups at the John Innes Centre, one led by myself and the other by Brandon Wolf. At Kobe University, the group led by Yukio Toza, and the University of Maryland, the group led by Vijay Tiwari. I'm sure we're all familiar with rice blast, caused by Pericularia oryzae, lineage oryzae, also known as Magnaportha oryzae. Less familiar, perhaps, will be wheat blast, caused by the same species, but a different lineage. This is a relatively new disease, having appeared in Brazil, in Paraná State, in 1985. It subsequently spread throughout South America, and unfortunately was accidentally introduced into Bangladesh in 2016, where it caused serious disease in farmers' fields. Originally, wheat, which carries the resistance genes RWT3 and RWT4, is protected from the various lineages present on wild grasses. For example, Avena lineage isolates carry both PWT3 and PWT4, which are recognized by the respective resistances in wheat. Lolium contains only PWT3, and this becomes very significant for the evolution. In the 1980s, the variety Anahuac became extremely popular in Brazil, and unfortunately, this variety only carries RWT4, making it susceptible to infection by the Lolium lineage carrying PWT3, which is not recognized by Anahuac. As I said, this variety was grown over a huge area, and inoculum buildup and subsequent mutation and loss of PWT3 has led to a situation where neither RWT3 nor RWT4 containing varieties are continued to be protected. Turning back to rice, many of the genes known to be responsible for rice blast resistance are of the nucleotide binding leucine-rich repeat class, NLR proteins. We merely hypothesized that RWT3 and 4 may also be NLRs. Previous work has already shown that both of these resistances reside on chromosome 1D in wheat. And as we all know, the D genome donor is Triticum tauschii. And furthermore, at the John Innes, the group of Brandon Wolf, you'll have already heard the talk by Sanu Aurora, have developed the AgRenseq diversity pipeline for cloning NLR genes. And they've done this primarily or initially for Triticum tauschii. We have a diversity panel, the AgRenseq pipeline. We approached Professor Toza's group, who had themselves developed fungal resources, in this case, isolate BR48, a wheat blast isolate, and others which they transformed to carry either PWT3 or PWT4. We combined these resources with those of the tauschii panel, put this through the AgRenseq pipeline, and held our breath, crossed our fingers that we would see something promising or positive. And indeed, we did so, both for foliar infection and for infection of the heads. We see this peak here in the Manhattan plot. This turns out to be a full-length NLR. The susceptible allele is in Chinese spring. But interestingly for us, the resistant allele is in cadenza. John is fortunate in having the cadenza tilling population such that we were able to identify lines carrying mutations within our candidate NLR. If this is the correct gene, we would expect them to show enhanced susceptibility on infection with the isolate carrying RWT4. We carried experiments both with BR48 and with the BR48 containing the additional effector PWT4. As you can see here, both on leaves and on the heads, the tilling line and the parent line are equally susceptible. If we turn to the infection with the isolate carrying the effector PWT4, we now see cadenza to be highly resistant. However, the tilling line has lost that resistance. So we have an NLR candidate, which we believe through many other mutations that we've observed to be a full-length NLR that controls the recognition of the effector PWT4 on both seedlings and head tissues. We tried exactly the same with the isolate carrying PWT3. We never found any clear phenotypes. At this point, we turned to a second diversity panel, again within Brander's group, this time made up of hexaploid wheat from the Watkins collection. And in this instance, we then found an extremely promising candidate for RWT3, exactly where we were expecting it to be on chromosome 1D. This is where Professor Tiwari's group came in, because they have a tilling population in the wheat cultivar Jagger, which contains the resistant allele of RWT3. So we obtained mutants from the group and tested these with the PWT3 isolate. And here we can see the Jagger parent showing the resistance that we would expect, and the line carrying the mutated NLR showing enhanced susceptibility. We turned back to RWT4 for reasons that I don't have time to go into, and were gratified and surprised, a mixture of feeling, to find a second resistance that also detects the presence of PWT4. And more gratifying still was the fact that this is homologous to the gene that we'd already identified on 1D. So to summarize, we have used various resources, the P-Oryzae isolates carrying individual effectors in a common background, to screen the Triticum tauschii DDM donor diversity panel and the Triticum aestivum diversity panels, combining that with agar-seq analysis, and revealed the identities of RWT3 and 4. In all instances, the 2 or the 3, depending on how you look at it, turn out to be nucleotide binding leucine-rich repeat NLR proteins. So we have now the capacity to identify within wheat varieties all those that carry RWT3 and RWT4, and we believe that this will be useful to protect varieties in areas where blast is not presently a problem, but where the environment may be conducive to infection by lolium and Avenia isolates. And also we've shown that NLRs play important roles in host range for the P-Oryzae lineages. So with that, I'd like to thank you for your attention, and BBSRC and Newton Fund for their financial contributions. Thank you.